decrease in dot 5, you have a smaller one. Okay, so we can have this intuition, but for age is a little bit different. If you have a 35 days low frequency stimulation that's around here, you decrease the age, you can enter the LTD region as you noticed before. However, if you have a high frequency stimulation that is already crossing the LTP region, and if you decrease the age, you're not able to reach this anymore. So for age, there's some counterintuition on how it will modify the orbits. And also some STDP protocols are insensitive to age changes, as I noticed in my simulations. So right now, we have a new plasticity rule that can unify key aspects of experimental heterogeneity. Okay, this new plasticity rule also it can reproduce experiments in 10 different papers. It is really nice. So it has certain robustness of the amount of curvature that it can encompass, and also it is also right now I'd like to make some conclusions of this new concept. So in the conclusions, we see that we can provide a conceptually new geometrical threshold. Right now, we're not talking about a single-dimensional threshold anymore, but a geometrical one. Okay, and we can see how sensitive it is to experimental conditions. For instance, if you increase 1 degree, you have this small shift. If you decrease 1 degree, you have this small shift. If you increase 1 degree, you have this different shift. Okay, and also you have an stochastic model. The stochastic model is much closer to the nature of dendritic spines that are very small. Okay, so this is not something that you see every day in plasticity models. And we can reproduce short-term depression. That's really important to justify how much resources your synapses are using. And this is really interesting as well. With the 10 experiments that we have, the 10 experiments that this model can reproduce, we just need one system of thresholds, just these regions. You don't need to modify the regions to have other plasticity outcomes. It's just one. So it means that somehow robust. Okay. But if you shake these regions, maybe you can break the model. Okay. And also, we can provide testable predictions. It means that we pass exactly, not exactly, but a much closer to the experimentalist needs to reproduce the predictions that we're making. So the experimentalists need to set the amount of calcium, they need to choose the age of the animal, things like that. And we can say, oh, if you use these parameters, you have much more chance of having these outcomes. So it can really test the predictions that we are making because you need to set this in the experiment. Another conclusion, another nice point, is that we can improve experimental design. For instance, it is hard for experimentalists to have LTD for old rats. We can propose a stimulation protocol to recover the LTD for old rats, for instance. It's something good that experimentalists may need. And for the last conclusion of this list is that we have the first plasticity model that can put together simultaneously the age of the animal, the temperature, and the extracellular medium. Okay? So age, temperature, and extracellular medium. Right now, let's talk about the bad points of the model. It certainly has some limitations. One of those is that it is not as fast as deterministic models because you have to spend some resources to make it more close to the appropriate level of description. So it's not as fast because you have to make simulations of the precise timing jumps. Another downside is that it captures only variability of physiological temperature. Because if you are doing experiment in room temperature, I cannot capture the variability of the data. And that's probably because I don't have the temperature effect in the release probability in the presynapse side, possibly. And also another downside, maybe people that are just want to have a very quick model, this model is too heavy, there are so many details, like it needs to put all the experimental conditions to run it. So if someone is naive, maybe, and naive in the sense that first time that's touching a plasticity model, you don't want to know about temperature, details like that. Maybe it's not so useful for them. So there are other kinds of models that do something similar to it. So for future directions, things that I would like to make with the model once it's a little bit more mature is to include other ages, because right now we have data just to reproduce from young to adult. We don't have the aged rat data to make a more broad model. And this is important because for the laboratories to keep very old rats is much expensive. If you can make a nice experimental design to know exactly what you will be recording for aged animals, you spend much less resources keeping them because you know exactly what you're going at. Another future direction is that we'd like to include diffusion, because right now our enzymes, they don't have diffusion, and we know that depending on the activation, some of these enzymes can travel between neighbor spines and can modify plasticity another side, another spine. And also, we like to include other dynamics, not only 2D, but maybe 3D to predict other synapse fates, not only potentiation or depression, but for instance, synapse loss. I'd like to know, for instance, what is the specific pattern that makes other things to happen? Almost the last is we'd like to include in your model modifications to simulate how pathological influences in the, for instance, the presynapse release mechanism will modify plasticity. Okay. In this case, I have the tau protein that is a very sticky protein that will bind to vesicles and will make the release probability lower. Okay, you can make this kind of implementations as well. And for the last of the last, we hope to make a generalization of this model to apply to other kind of problems, not only plasticity, by detecting, for instance, to creating a rule that says this orbit likes to spend more time here than there, and creating like a classifier for different problems that takes orbits. So our last comment. Right now, in this year, we submit the paper to Nature Communications. Right now, this paper is under review by peers, and we are waiting for replies, but it's a good thing, I think, a good sign that our paper was accepted there. And that's all. That's all. Thank you very much.
Thank you, Yuri. You're slightly over time, but that's very interesting.
Okay.
That's fine. So we're going to get into the second part of this defense, i.e. the session of questions and answers from the committee members. And I will start with the reviewers. And I'll start by asking Sarita Nadkarni if she has more questions or if she wants to make comments on her report.
Hi, Yuri. Very nice presentation. Huge amount of work, and I really enjoyed your phase space analysis. And I guess you could increase the dimensionality of your phase space analysis, right? So I think in that context, I have a bunch of philosophical questions and maybe one technical question. You haven't included sizes and presence or absence of ER.
Of what?
Should I let you answer that first and then?
You said that I didn't include sizes of the spines.
Sizes of the spines, presence and absence of ER. And for example, something like neurogrenin could also further modify plasticity profile and or further modify CAMK2 and
yes.
Calcineurin. So have you thought about how you did amazing work in the sense that you're able to get consensus with 10 experimental results. but there's bunch out there, right? So how do you think this work fits in with, do you think that you just need to add more and more layers of sophistication and detail to the model? Or do you think there's a better way to do it because there are several other sources of variability?
Yes.
So sizes first, sizes and ER, depending on the spine sizes, some of these experimental results might not convert, right?
Well, we have sizes of the spines, but it's not a 3D structure. We can set the volume of the spine, and this will modify the calcium influx, right? Depending how much volume you have a different concentration because you have more space to fill when the calcium enters. So the volume we have, we can play. We know that when you are starting to model KMK2, we had the data together with this signal of KMK2 that's showing the transient change in the volume. And we know that if you have a small increase in the volume, you have a small decrease in the concentration. And then we try to model this in a very rough way, and we fail because we don't have too much details about how the structural proteins will be activated for different stimulation protocols. So we didn't keep the volume change in the model. But about your second...
I understand that you might have explored it, but in the context of the source of the variability might also be differences in sizes and presence or absence of ER. And so how do you, and this is sort of a more philosophical question, right? And there are other, I guess, sources of variability. For example, astrocytic feedback.
Yes.
And so how do you see your model sort of fitting into this whole messy can of worms?
Yes.
The reason to ask you is because I think the agenda was to make sense of this, what seems to be sometimes an apparently data that is contradictory to each other. So are you happy that you've taken care of certain data sets and that's what one can do, or
I see,
do you sort of see this?
I see. Well, there are two possibilities. Okay, one, it is we have the model that's very complicated. We don't have everything there.
Right.
We are missing ER that's important for plasticity maintenance. We are missing a lot of other enzymes. But there are two possibilities. One, you increase the model, putting more things on it, okay, and you have to make a new interpretation of what is the activity. Another possibility also is to decrease the complexity of the model to be able to put it in a dendrite with many different spines, for instance. So I think I am missing a lot of things in terms of, because there's a huge complexity, but I'm trying to be as close of the data as I can in terms of the things that I included. So the things that I don't have, for instance, ER, I am assuming that they are working there in the background, totally nice. But if you want to make it explicit, I think it will be much complicated, much harder to make it. You see what I mean? But also, there's another thing. If you remember a part of the thesis, I was showing different kinds of models and what they cover. Right? I was showing these guys, they cover temperature. They don't cover temperature, but they cover very nicely. They cover at least reasonably the astrocyte. So I identify there's a lack of plasticity models that cover certain aspects. And the model was done to try to fix these things. And probably they can be combined or not. It's something that I need to try.
Yes, you're right. A model should not even try to achieve all the details, right? But do I have time for another question?
Oh yes, sure. You have time. Yes. This is
so I guess my second question is slightly more technical. The PDMP that you spoke about, and you had certain discrete variables and certain continuous variables that kind of progressed in tandem, right? So I actually was hoping that you'd be able to tell me why did you not choose all of the variables? Why this combination of continuous and discrete? What was the optimal choice
Yes.
of discrete and continuous variables?
Yes, I can say it was the speed. Okay, one aspect was the speed, and for instance, we had the SK channel that it opens even if you are in resting concentration because you have the calcium flickering in the background, and so since the SK channel is sensitive, it will be open all the time as well. So we had to remove some stochastic components because you take too much time measuring the jumps. So this will slow down the simulation. So you had to choose what is the ones that you really need to understand the calcium dynamics, and which one maybe you can put it deterministic.
So when you're going back and forth, all the variables were getting updated, wherever the update came from, there was this tandem updating of all the variables. Is that how it happened?
It's not like that. It was not like turning off one, turning off another. But I had another model that was fully deterministic, is in the thesis, and it was slightly different from the one that was half stochastic that I just presented. And the variable that made the great difference was the voltage gated channels, because it's open all the time. So this channel, since it's open all the time, not like AMPA receptor that's just in a single event, I really needed to make it stochastic because it was making a lot of difference. So in the end, I had some channels that are stochastic, like the VGCC that are important for the baseline, and I had to remove other ones like the SK that are just making a lot of jumps, and there is not much difference.
But the SK channels can be a small bunch, right? So they can also be a major source of noise.
They can.
But did you test? So you probably tested. You probably tested this to make sure what are the major sources of noise, and then decided your discrete variables as opposed to continuous. So one last question, Yuri, and I think I'll pass the baton to somebody else. Have you thought of incorporating presynaptic long-term plasticity? And if you were to do that, how do you think your results would change? And your take-home message for experimentalists before they put out another data set on plasticity, what is it that you don't want them to do and what is it that you want them to do?
Okay, that's a good question. Well, to include long-term changes in the presynapse side, you can hear me?
Yeah, I can hear you. Yeah.
Okay.
Can you hear? Yeah.
So I had to include what is the name? The pathway of I forgot the name of the pathway.
Synaptotagment, did you mean?
Yes. So I had to include them and have to retune the model to see how the changes in the presynapse side will affect the whole orbits, the whole dynamics. Probably there will be a change in the regions if I do this tuning. And you had a second question, Suhita. I'm sorry. That was
experimentalists. What's your take home?
Okay.
So that
my take home for them. Well, I wish them to make experiments that are a little bit closer to physiological values. Okay, like for instance, physiological temperature, physiological calcium. It would be very nice if they do it in vivo, but it is extremely hard to control exactly what you want in vivo and to probe everything in vivo. It's also, even if you do in vivo, you're not exempt of changing the environment. If you stimulate really fast, also you can modify the temperature because you can make the electrode to heat and will modify what you're doing, what you're recording. So I think what they can do is try to agree to each other to use the same experimental parameters. But I know that's extremely hard to, because then if you have the same experimental parameters, it will be easier to compare between their outcomes. That would be ideal, but I know that's impossible because every laboratory, they have different materials, resources, and it's really complicated.
Okay.
But it's hard to agree.
You just have to learn to live with the mess. Thanks, you.
Thank you, Sufita.
Are you okay? You don't have any more questions?
For your future endeavors.
She's gone. Okay. All right. So no more questions from Sufita. Now, Nicolas Brunel, you were the second reviewer. Would you like to ask more questions?
Oh yes, thank you. So first, I'd like to say that I really enjoyed this oral presentation. I think it was very, very clear, and also I greatly enjoyed reading the manuscript. I think there was a really very comprehensive description of the literature, and I was quite impressed by the amount of work that you've done on this model. So I have a number of questions on some basic features of the model, and the first one is these two regions in the CAMK2 calcineurin plane. And my question is whether these regions are consistent with pharmacological experiments so people have shown that if you block CAMK2, basically you block potentiation but not depression. And vice versa, if you block like calcineurin, you block depression but not potentiation.
I see.
And so to me, that doesn't seem compatible with naively, so just from these results, if I had to sketch regions in a CAMK2 calcineurin plane, I would sketch regions in a very different way, right? To be consistent with this data. So I'm just curious whether.
Okay.
So is there something that I'm missing? First, did you try to see what happens if you just block CAMK2 or calcineurin, and what happens? And so this is my first question.
All right. Thank you, Bruno. Well, I had a try with O'Connor data. I don't know if you remember this data set. It's a frequency-dependent stimulation that they saw like a BCM curve, and they block differently. They inhibit calcineurin and CAMK2. So I tried that. I managed to reproduce the control data set, more or less. Okay, not for very high frequencies, but when I try to inhibit calcineurin, I don't know exactly. So the answer is I don't know. Okay, but what I saw is that when I try to inhibit calcineurin, it happens that the CAMK2 will be overreacting, right? So the orbits for calcineurin will shrink, and the orbits for CAMK2 will reach the saturation much faster, okay? So the thing is, I don't know exactly how much of the inhibition will modify the kinetics. Okay, I don't know if the reactions of the CAMK2 substate will be modified and how. If I had the imaging of how they will react to inhibition, maybe I can try to model it. But there's another aspect as well, is that when you inhibit these enzymes, you probably, I'm not sure, but you probably have some compensations of other enzymes, okay, or some kind of compensations. And the model does not have explicitly this thing. And I believe that's a good point of bigger models. You have other pathways that are recurrent. How can I say that? They are repetitive. So with these other pathways, maybe they can compensate a slight change in one enzyme. But if you have a very simplified model, you cannot see these compensations, and everything will be up to the other variable. So a block experiment in my model didn't work for this, for CAMK2 and calcineurin. Probably a bigger model will be able to make a better inhibition blocking experiment, but also they need to see the data and how much this will affect the reactions. That's all.
Mm-hmm. Okay, so your guess is that basically, if you would add more of the basically the biochemical networks in the PSD and more elements, then maybe that would help in reproducing this.
I
this how
believe.
much could explain. Yeah, that's plausible. Actually, so related, since we're talking about the key players in plasticity. So there are a number of already published models that describe the dynamics of CAMK2 autophosphorylation, and in many of these models, people have included another phosphatase, PP1, which was kind of that dephosphorylates CAMK2, and here you chose not to include PP1. So I was just wondering if you could explain the rationale for ignoring PP1 and including only calcineurin as the only phosphatase.
Okay. Well, we'd like to have something very similar to what John Lisman had in the initial paper, that was calcineurin and CAMK2 in modeling. And also we choose to not have any reaction between calcineurin or phosphatase and CAMK2, because in 2015 there was a paper by Otmakov and Lisman together, that was the guy that made this hypothesis, that they show that no phosphatase modifies the CAMK2 decay time. So they are suggesting in this paper that if you inhibit every phosphatase one by one, all known that all phosphatase they are able to inhibit, they don't see any difference in the CAMK2. So that's why I don't put any reaction between calcineurin and CAMK2.
Okay, I see.
Satisfied?
Yes. Yeah. I have actually another question about the calcium control hypothesis. So one of your main arguments for building a model that's now two-dimensional, essentially, as opposed to being just one-dimensional with just calcium, is this data from, if I understood correctly, you have this data from Tigares experiments showing that you no longer have plasticity at really high calcium. Yeah. But to me, that's not really that really does not invalidate calcium control hypothesis. That just invalidates one form of calcium control where you have potentiation whenever the calcium is above a potentiation threshold. But you could certainly modify this hypothesis and add a third threshold, like a higher threshold above which you have no plasticity. So in other words, to me, to really invalidate calcium control hypothesis, you should show that for a given level of calcium, you could have different plasticity outcomes, that the plasticity outcome is not only controlled by calcium. And I'm not sure that Tigares experiments really show this.
Yeah, I agree with you. I agree that it does not invalidate. And also there's another problem about, I agree that we cannot throw away the calcium control hypothesis just by the amplitude. First, because we cannot measure very well the calcium, because when you are measuring, you are modifying with the laser, with the buffer, because you decrease the diffusion of calcium, it makes it more slow. So when you are measuring the calcium amplitude, you cannot have a very nice comparison. And also there's another problem related to measurement, is that for you to measure the whole calcium protocol, you need to use a lot of laser, and this will surely modify how everything works there. So first, we cannot observe very well. There's another paper by Nivian and Sackman, the guys that won the Nobel Medicine. They also show that you can have the same amplitude of calcium, and you can have LTP and LTD, like totally opposite. But the issue is that you cannot, you don't measure the whole calcium signal to know exactly what is going on, and also you have this the dye making it harder to observe. So yes, I agree with you. Maybe you can modify the calcium hypothesis to accommodate what happened if you have a lot of calcium. But so far, I think it's hard to go for this direction. And also, if you make a 2D, like instead of 1D only, if you use two dimensions to encode plasticity outcomes with calcineurin and CAMK2, you have more space to separate the protocols, so you can have less rules to change, to try to incorporate many experiments. You see, I think that's my answer.
Yeah, I agree, and I agree that also the paper by Nevian and Sackman is I think is a stronger they have stronger arguments against the calcium control hypothesis. Though I think you can still, I mean, it's complicated because
You can fix it probably.
yeah. So I think one crucial point is that if you just look at the amplitude of the calcium transients, you might miss. I mean, it really, in the calcium control hypothesis, at least in some versions of it, well, it's more important the time you spend in specific calcium regions as opposed to the amplitude of the transient. So
Yeah.
I think that makes things more complicated.
Sorry.
Go ahead.
Okay, no, there's one thing is that many models that try to predict plasticity, they pass the signal to some slower variable. And if you have a slower variable, you can integrate better what you're doing. If you have already an enzyme that is doing this job of integrating the very fast signal that's calcium, that's not so fast as voltage, but it's still fast, we can try to see what exactly is going there. Because if it's fast, like calcium, maybe it is hard to separate the different patterns that will induce different plasticity outcomes. So I think the calcium hypothesis is nice because it's easier to record than enzymes, for instance, right? It's harder to record in voltage, but it still is more accessible than they have to design a specific sensor for enzymes. So it is interesting if you can develop a theory to observe just calcium and have plasticity predictions. But I think it is really hard because it is too fast. That's my hypothesis. The calcium is too fast to be used efficiently.
Yeah, I mean, I agree that there needs to be some slower integrators downstream. I think that's a reasonable assumption. I have another question on the model. So your model is in the spectrum from really highly simplified to very detailed. So your model is quite detailed. So, and in fact, I think you did a really good job at really modeling all the components. I really liked how you built step by step the details in the model. But as you said at the end, there is a drawback that it's actually it's a pretty computationally expensive model. You have lots of variables. So have you thought about kind of approximation methods that could, for instance, you could imagine that instead of describing populations of like large populations of channels, there are approximation methods that you could approximate it by just a single stochastic random variable that could match maybe the two first moments or things like that. So have you thought about going in this direction to try to simplify the model and make it
Instead of
faster to simulate?
Instead of using the discrete simulation, use like an average of the channel activity. Well, I didn't try that. And actually, this is a project that's ongoing after my PhD for a master's student. He's trying to simplify my model to be able to escalate it up. But I never tried. I never tried that. But I'm not sure if it will work as well, because maybe the average will be not exactly on the activity, for instance, of a T-type channel. It maybe the average will be right in the middle, and not taking the precise opening and closing of it. Maybe these discrete events matter a lot. But so in fact, I don't know. I need to compare to see, but I'm not sure if it will work because it's outside the real value of it.
Yeah, I mean, I think you can still. Well, I think it's an open question whether clearly it's important. What you showed, it's important to include the noise, and also it's really nice that we can reproduce the variability in plasticity outcomes, which is really a very prominent feature of basically every synaptic plasticity experiment. But maybe you don't need to have such a detailed description of at the channel level. Maybe a more simpler description of the more. But yeah, it's a
maybe like Sugita mentioned, maybe I can try to shut down the stochasticity of a single channel each time to see how different the curves will be. But the thing is, I did all at the same time. Like I put a whole deterministic against a whole stochastic, and the difference was really small. It was really small, and the difference that they highly impacted this difference between the orbits is the T-type channel that is always flickering, and maybe it states that stochastic is really important to have it.
Okay, well, thank you. I don't think I have any more questions. Thanks a lot.
Yeah, thank you, Nicolas, for your questions. Let's go to the examiner, Kim O'Donnell. Did you care asking questions to the candidate?
Sure, yeah. I'll at least after he finishes his drink, I'll start with the first of all, well done, Yuri. It was a pleasure to be involved in the project and to work with you over these years. And like Saida and Nicolas said, it was an excellent presentation. I found it very enjoyable to see the whole work from review from your point of view, and I thought you gave a very coherent sort of philosophical argument for the project. So my question is, in your presentation, you framed the sort of output of the model as a plasticity rule. So you're kind of saying, I don't know, something that captures the relationship between the stimuli to a synapse and the synaptic strength change. But the simulation is quite detailed, and the rule really isn't explicit. It's just implicitly embodied in this very detailed simulation. So I guess my question is, what is the rule, and how do you think we can use how would you suggest we use the insights from whatever that rule is to understand brain learning? Like what's the implication for theories of brain learning?
Yeah. Okay, that's a very hard question. I will try. So what is the rule? Can you hear me? It's a bit.
Yep. No, I can hear you. Yeah, sorry.
Okay. What is the rule itself, right? Because you say you have a Nielsen plasticity rule, but what is the rule? For me, the rule of this model is how you have the dynamics spending time in the different regions that they call the hotspot. So for me, the rule is this. You have this hotspot and you have another hotspot. And what is interesting, that every experiment that I tried, that were many, they spend the time in that specific place and they share this pattern of activity in this map. And the thing that maybe can be extended to other ideas is that maybe you have hotspots of activity regardless of other parameters, other things that can affect the experiment. For instance, you have a hotspot for LTP right here. And maybe you have a hotspot that says that your neuron will die. Maybe you have a hotspot of activity of some enzymes that says that you have a maximum growing of your dendrite. So probably you have these hotspots. Not only thresholds that after the threshold everything should be like that, but a hotspot in a higher dimensional geometry.
Now, hotspot meaning it's a bounded region. Is that what you mean? It doesn't just have a threshold and then everything beyond it?
Yeah, but not necessarily a bounded region. It have to be enclosed, but maybe can be two different for the same thing, but they are separated. Okay. So the idea is this one.
And okay, yeah, that's a fair summary. But so, what does it tell us about brain learning? Like, how can we use this to understand how the brain learns better?
How we can understand that? How can we use that to understand how brains learn better? Well, one thing that could be interesting, for instance, is for in vivo recordings that you have much less control of the parameters and experimental conditions. You can, for instance, to just using the spiking times to simulate what will be the plasticity outcome, because sometimes it's really hard to have patch clamp in vivo, for instance. So just if you know the spiking time of the two connected neurons, that's already really complicated to get. In vivo, maybe you can try to simulate the plasticity outcomes. I'll give you an example. For instance, I was trying to make a model to see how sleep will affect the downscaling of synapses that you expect to have long-term depression during sharp wave ripples. However, it's really hard to have the times of two neurons that are connected in vivo. So you need to record them when they are sleeping or when the rats are sleeping. However, it's really hard to know when the two neurons are recorded, are connected. So using the model, I believe that we can have a shortcut to guess what is the plasticity outcome in vivo situations. And this is really complicated to do with other rules because you don't know exactly the experimental conditions, the temperature, and we know that in vivo, that's something that you want to learn, how the brains will be, how the synapses will change. I believe that we can use the model to have this greater understanding.
Okay, as a tool for predicting quantitative changes, something like that?
I'm sorry?
You would use the model as almost like a black box tool for predicting synaptic plasticity changes?
If it could be used like a black box.
Well, I guess, sorry, the black box part is not so important, but you said you would use it as a tool for predicting.
Yes, I believe it is possible to have a nice idea what's going on in vivo. Yeah.
Yeah, that's okay.
Yeah.
Yeah. And
this is important.
Can I ask another question?
Sure. Okay.
And so it's related to Nicholas' questions about the calcium control hypothesis. So it's true that your readout mechanism is 2D, and the classic calcium control hypothesis models are sort of one-dimensional. But in this model, the CalK2 and the calcineurin are downstream of calcium. So whatever signals they're sort of representing or demonstrating is inherited from that 1D variable, the calcium. So it somehow feels inherently, does that not make it inherently 1D? And you've just sort of, those two variables are, projecting that 1D signal to 2D. So why does that, you can't create information by projecting stuff to higher dimensions. So why would the 2D readout work better than a 1D one?
Because you have more space to separate between the different protocols. If you have a 2D, you have more space to cut exactly what is the difference. But I understand the question that why you don't interpret calcium in the source instead of looking in the cascade. But I believe that the problem for that is because, as I said before, it's really fast, and because it's really fast, it's hard to measure. And since if you just look in 1D, it will be much complicated to make a nice cut to separate what is one thing or another. If you have more variables, a bigger space, you can have more space to separate things, so it will be easier. Even if it's the same information, but it's better organized.
I see. Okay, yeah, that makes sense. Okay, thank you.
An idea which is very much a very large use in AI analysis of large dimensional data. They do that all the time, projecting higher dimensional spaces.
Yeah, I guess we do.
Actually, part of the inspiration for having a high dimensionality comes from the idea of the curse of dimensionality from machine learning, that states that if you have a bigger dimensionality, you'll be at least the distance between your points will be at least bigger than 0. So if you have some even a not meaningful variable, you can improve your separability. If you have a variable that's hard to separate, you can add noise and improve the separability of the variables. So even if the KMQ2 is totally saturated, it's hard to separate because they're overlapped, it means that you can add it, and if it has a physiological meaning, and improve the separability. See what I mean?
Yeah, I mean, seems surprising, but I believe you.
Are you satisfied, Kian?
Yes, thank you. Okay.
No more questions?
No, I have some small points, but they're the only.
Sorry, I didn't. So you're happy with your question?
Yes, I'm happy. Yeah, thank you. Sorry.
Happy. Okay. Well, I guess before I give the floor to the two advisors, I would like perhaps to thank you for your very nice presentation.
Thank you, Olivier.
And the document as well, which is well written, well rewritten in some cases, but that's fine. I have a number of burning questions. One of them is about the, well, this is partly explained in the thesis, but could you tell us more about how you tune the parameters of the model? This is probably...
I can tell you.
Sorry?
I can tell you.
Yeah?
With a lot of time, I had to do it manually because the system is slow. So I had to move these regions. After having the simulation, I have to move these regions to the nice place to take all the experiments at the same time. So that takes a lot of time to know exactly where the points of the polygon should be. And after having a lot of trouble trying to tune these regions, I decided to make a deterministic version of it to run faster, to see where I can put the regions. So having this stochastic model, I made this deterministic version. It helped me, it accelerates how fast I can change the polygon position to know exactly where to put it. But it took me a long time. I had to do it manually, and it was not maybe the cleverest way.
There was no way of doing some sort of optimization?
Probably.
Perhaps, but you didn't try?
I didn't try. Probably there was a way, but you had to wrap everything and know exactly what is changing. But also there's value in not doing automatic optimization because you know exactly what each variable is doing. Yeah. If you are moving things without seeing it, maybe you are missing an interesting detail about the system. So there's value of changing. It's slow, it's painful, but you can understand better your system.
So you must then have a very accurate idea about how sensitive the model is to the choice of this and that parameter. Which are the most important parameters in the stability of the model? Okay,
for prediction, the position of the boundary is really important.
The prediction?
For plasticity prediction, the position of the boundary is really important. I tried to make a diffuse boundary once, like instead of a fuzzy one, and it didn't work as better as the instantaneous one. And also another parameter that's really important is the calcium permeability. It modifies how the calcium, how strong will be the calcium entering. This is also another strong parameter that I use like almost as a free one.
So geometry is very important, you said?
Yes.
Geometry of the regions of the polygons, and then this parameter.
Yes, these two parameters are really important. And also there's an issue with not issue, but we need to take into account how fast the enzymes will saturate. Because if it saturates really fast, it hits the limit, and the other enzyme will have a push. Mm-hmm. Because once you saturate something, everything will be concentrated in the other enzyme. So it needs to be in a way that the first spikes do not saturate everything at the same time. So we had luck with the data because the data tell us how fast it would saturate. Yeah.
I would like also to ask you, perhaps once again, maybe I asked you the question yesterday during your talk, the role of your noise, the noise in the model. So there is noise in the model. How important it is? I thought I heard you say that it's not that important because the deterministic model is almost as accurate. Am I correct?
It is almost as accurate. It is faster, almost as accurate. But there's a detail. The most important noise that we have is the presynaptic one, because it's all or none, right? It will have or not. In some models, they use averages of this all or none release of vesicles. For instance, you have every release, you have 1 micromolar of glutamate, okay, and or 0, right? 1 or 0. So in some models they use an average of 0.5 all the time. They don't have the extensive short-term depression that you have in your model. And this, if you have an average average of release of glutamate, it will make a lot of difference in the geometry.
In the geometry.
In the geometry, because if you have an average, in the long term, this average will never really reach the 0. It will be always contributing a little bit, and if with a discrete release, you have 1 or 0, and sometimes only 0 for a long time. So this small contribution of the average will kick the enzymes really fast, and this will saturate both sides. So one of the most important stochasticity is the presynaptic, and later is the baseline that makes the small difference between the deterministic version and the stochastic version. Yeah. Okay.
And you didn't find it useful to, so you have PDMPs forward. So in between the jumps, it's an ordinary differential equation.
Mm-hmm.
And did you try to see whether using an SDE in between, stochastic differential equation, would make any difference? Or
it didn't
make any sense, or it wasn't useful, or you didn't try it?
I didn't try it.
You just didn't try
Yeah, just continuous ones.
Okay.
Yeah. Just continuous.
Well, that's the regular PDMP, yes, of course. Yes. I'm not sure you told us exactly where your model fails.
Fails.
What is it?
I told in the.
Well, you said a few.
Yes, I can go back there.
Yeah.
Like.
Well, you did. You had this one slide.
It was very, very short, right? I don't want to show much of the failures.
Yeah.
But one thing, how it fails is that it does not capture the variability of plasticity outcomes when the temperature is too low.
Yeah, I remember that.
And I believe it's because I don't have the temperature implemented in the presynaptic release. That is the most important stochastic trigger. So
you insisted on this
Yeah.
Temperature business, but anything else where?
Where it fails, for the blocking experiment, for instance, I cannot. I don't know exactly how to block the KMK2 or in calcineurin. It breaks the system because you cannot compensate. Where else it fails? If I try the experiments in mice, it does not work all the time. I had to make too much changes that are not exactly as the experiment, because mice and rats, they are very similar, but the plasticity outcomes may not be. Yeah. So I had one experiment that I reproduced that is from mice, and I had to change the magnesium a little bit. It's not exactly as the experiment. So I believe that making a model only for rats, only for hippocampus, I think is already quite complicated. And probably it's possible to modify it, but to other regions, other animals. But that's how it fails. That's the limitation of only.
It's not very much portable so far.
You need to be modified to include other things. Like, yeah, probably you can. For instance, I did for hippocampus. Maybe it's easy to adapt for cortical plasticity. For instance. There's a lot of experiments to do that.
Okay.
What else? I think I don't remember other failures.
Yeah, that's already good answer. Also, several times, I think in your answers to one of the reviewers or examiner, you mentioned the fact that you had to use time scales. Or something was very fast and something was slower.
Yes.
So that immediately raised the flag in my head. Slow, fast systems. You've been spending part of your PhD in a research project.
Yes.
whose goal is to understand and use slow, fast systems in neuroscience. Did you ever use this theory or think of using this theory, or do you think it would be useful?
I never had. I never imagined to use it. Okay, but I talk with people that studies. My colleagues, they had to hear many hours of me talking about this model, and they study fast-slow dynamics every day.
Yes.
And they never told me, hey, you know what? Maybe you should be using fast-slow dynamics here. Yeah. But maybe it's possible that but then you have to make it analytical but treatable. You know, have to bring it to remove the stochasticity maybe to simulate faster,
Okay.
or maybe not. I don't know.
Okay. Well, thank you. Let's see. Well, I think I've emptied my bag of questions here, and so thank you very much for the answers. I would like to give the floor to your two advisors in sequence, not in parallel. And the first one is going to be Romain. Romain Vels.
Okay. Well, Yuri, I'm not going to ask you any questions. I'm just going to start by saying to congratulate you on the defense and the presentation. I really like the way you selected a few points in the model and highlight them. I really like this. Overall, I think we were very fortunate advisors with Hélène, because you agreed to embark on a very ambitious project at first, which is to design a new model of synaptic plasticity. And knowing the amount of data and established models, it's not an easy decision. Also, I think we are fortunate because after being, let's say, blocked for a long time on this tuning, finding this new, let's say, synaptical or conceptual 2D plot unlocked a lot of discoveries, and it was very exciting moments. And as a researcher, I can tell that we don't have these many moments in your overall day-to-day researching activities. So this was a very exciting time, and it unlocked a lot of possibilities. I think they haven't been, by lack of time, they haven't been studied. For example, studying how the model react when we have like young synapses with a small number of ampere, things like this, which are very exciting questions. And also, I have to acknowledge that you choose and you agreed to write a big paper. And as a PhD student, this is difficult because you end up with a single paper, maybe not, which is in review, instead of having many small papers. And for that, I would like to thank you with Hélène, because this is not a very nice, this is a very hard decision to make for a PhD student. Overall, I think the years on my side, they went very fast, and I enjoyed a lot all our discussions, arguing about whether we should put details, remove details,
it was nice.
Put more concept, remove concept, and like Surita said, this is not very easy to strike a balance. And at the end of the day, this is you have to say to deliver a model. So it's not that easy. So with this, I would like to thank you, and I would say to conclude that overall, I really enjoyed the time I spent with you. And,
Thank you, Romain. And Hélène, would you like to say something?
It's my turn, Yuri. So it's been a very interesting journey for me. This is not my first. I'm a biologist, as you know, and you could hear this every day. Well, not every day. Whenever we had time to speak, it was my biological point of view, and I was surrounded by mathematicians and computational scientists, including Kim and Romain. So it's been very interesting, and I think we were very happy to find you, because you were really in that middle ground where you could understand me and you could understand them, and I think we journeyed together quite well. There were some ups and downs. I was very, I like the question of Nicolas about this point that, you did point out that most of the models did not allow to explain why with too much calcium there's no more LTP. And I was bugging you guys on this because I had this data in my lab showing this published, and it was counterintuitive. And I bugged you about the escape channel, I bugged you about all these things, about the safeguards, and now I see them, and I think this partly explains why we have these issues in biology, and I really hope that this model, which I'm very proud of now, I know I have to congratulate 3 of you more than me, because including Ken, because you really worked very hard on this, and I was just trying to keep the biological boundaries on this model. But I think it could be really useful for us as experimentalists, and I really hope that we will be able to sell it and to make it more available to people like me and my team, so that we can really gain a lot of time and effort in terms of working with animals. As you know, this is really important for us nowadays. In terms of supervision, I have to admit that this was mainly Romain's work. And the fact that you got a big paper, he's right, it's difficult as a PhD student to just go for one big paper. But you will see that this will pay off. It has to pay off. This is, in my lab, this is what we try to do. It doesn't always work out. But, at least in biology, I don't know how you work in mathematics, but in biology, we'd rather have one very big influential paper than a lot of small papers. So I really think it's going to pan out, and I really hope you're going to do better. Better, it's going to help you do a great career afterwards. I was a little bit tough at the end, as you remember.
It was very complicated,
but
it was...
you stick to the deadlines. This is my role, and I'm sorry if that really hurt you, but I don't think it did. I think it just drove you a bit too fast, maybe. But
flying.
But now here you are today with the PhD hopefully concluded. And now the next step is to publish this paper, and I'm really looking forward to the peer review. It was very hard to knock at the door of the editors. I've had a lot of experience with this, so I know I was telling you it was hard, and I'm really hoping we're going to get there, and it's going to be successful, and we'll have the time and the effort and the energy will pay off. I also want to thank very much the reviewers and the rapporteur. I think they did a good job. The questions were very interesting. I think Romain and I and you and Kin have to discuss this, some of them, because they are probably going to be part of some of the reviewing that we're going to get. And so the next step is not finished, but we'll be there. So I look forward to the paper, and I really wish you good luck for the rest.
Thank you very much.
Thank you, Hélène. Well, I guess that finishes the first part of the defense, with the second part of the defense. And now you're going to have to leave the room, and we are going to stay here and discuss the final details.
All right. Oh, this just water. Thank you very much. I'll let you to have a discussion.
Go get a big drink, Yuri.
All right, so, well, thanks to the three of you for having sent to